Well, first of all, please let me thank for putting this together, and for giving us the opportunity to give you an insight based on what's going on in our lab right now. It's going to be a shared presentation, so it's going to be me giving you a bit of background about what's epigenetics and the knowledge about epigenetics in marine invertebrates, specifically in corals. And then Javier is going to tell you a bit more about our specific work when it comes to studying those mechanisms. Well, using one speech. Well, Javier will tell you one. Well, so the work in our lab at FIU, the Marine Sciences Program of Florida International University, it's interested in studying environmental genetics. That is, how organisms respond to the environment, not because of their information on their genes only, but also because of certain epigenetic mechanisms that are going to modulate how genes respond to the environment. Now, I don't need to tell you how important is global change that in the current, climate change scenario, and essentially that is probably a major threat, the most important threat for oceans worldwide. We are interested in studying how different drivers resulting from global change affect marine invertebrates. More precisely, in the case of corals, we're interested about studying the role or the effects of temperature and nutrient pollution. Now, when we look at the given habitat in the ocean, such as coral reef habitat, and we look at how changes in temperature or increasing in nutrients in the water, how those are going to affect those organisms, well, we need to think about different levels. We need to think about populations, communities. We need to think about individual organisms. We need to think about symbionts, and also we need to think about the most fundamental level in those organisms. That is the genetic material. There is where our work mainly lies. We are not just, most of our work, unfortunately, is not going into the reef and dive. We're definitely looking for every opportunity to do that. But we are mostly based in the laboratory, the pipettes, and doing weird stuff with DNA. So essentially, our interest is precisely that, is, studying first DNA, sequence of DNA, genotypes, but not only that, we're also interested in studying epigenetics. So epigenetics, the definition, the formal definition is the inheritable changes in gene expression that are not due to the sequence of the DNA. So you must be asking yourself, so what is then behind those changes, those epigenetic changes? So there's where the environment comes in. Okay? So essentially, those epigenetic changes are, for instance, this is a very classic example of phenotypic plasticity in the case of the water fleet duck. That depending on the presence or absence of predators, they're going to change completely their phenotype, right? So how is it that if these two guys are genetically identical, they can be so different in terms of phenotype? How is it that a cell in our body can be a brain cell or a muscle cell, completely different phenotypically, functionally, but at the end of the day, genetically, they are both clones? Well, that is thanks to those epigenetic marks that are going to activate or inactivate selectively different genes, right? Now, our interest is to study epigenetic mechanisms in marine invertebrates for two reasons. The first one is because there's nothing known about those. And although, fame and fortune of epigenetics has been built on health, on cancer, on disease, on human health, right? But, surprisingly, and when we're mentioning so often the word environment, nothing has been done so far when it comes to ecologically or environmentally relevant organisms. So that's the first reason. The second reason is that we believe that by expanding that knowledge into a broad range of organisms, we're going to be able not just to understand how the cells or their epigenetic mechanisms work, but also we are going to be able to provide answers for current challenges. And I will tell you a little bit more about that in a few seconds. Okay, so crash course about epigenetics. Just picture genetics as the hardware in your computer. You have a computer with a certain amount of memory, a certain amount of inches in the screen, and then you have the software. The software is going to be either Windows, or it's going to be Ubuntu, or it's going to be Apple OS, whatever, right? The thing is that is epigenetics. That is essentially what's going to be providing or depriving from access to different parts of your hardware. Okay? So, the beauty of epigenetics is that those marks, different or oppositely to what happens with the genes, are reversible. They can be put there or removed depending on different signals. And those are mostly environmental signals. So you can sort of use your software to open your mail client, and then you can close it, and then you can open some other program that you can make your computer into. It's going to turn it into a movie CD, right? So this is more or less the same. But behind all this, probably the most important feature of these marks, that can be like many different names, is that they can be inherited. And that is the key word here. Many people identify epigenetics only with things that can be transmitted to the offspring. Start thinking about corals and how they can transmit these phenotypic changes to offspring. But these marks can actually affect how different cells are going to behave during, let's say, the course over a single life, right? So anyway, at the end of the day, you're probably familiar maybe with DNA methylation studies, which are probably the most abundant and famous when it comes to epigenetics, but also some other mechanisms involved in structural proteins in the chromosomes, as well as small RNAs. Anyway, all of them have been demonstrated to be able to be transmitted to the next generation. So, all in all, and I'll start talking about corals right now. Wait. So, all in all, this constitutes what's the basis of environmental epigenetics, which is, okay, let me look at what's going on in the environment. Either it is ultraviolet sunlight, or either it is nutrient pollution or temperature. Now let me look at the epigenetic marks, and most importantly, let me look at how that translates into a physiological response into the organism. Okay? So that connection is the basis of environmental epigenetics. Now, coral reefs. Okay, one of the most important habitats worldwide. Nutrient and thermal stress are probably among the major threats for these habitats. Now, we know that things are, like in these cases, bleaching is going to be the result of increases in temperature, and what we know for sure is that corals are going to survive those conditions only or based on the ability they have to respond to those threats, right? Now, I want you to start thinking about trying to make those connections of what I just told you about the genetics, the epigenetics, and these environmental threats. Now, the ability to respond is going to depend on many different things, because corals are extremely complicated beings. Now, the thing is, we have the genetic constitution, the hardware of the coral. We also have something that is called the microbiome, which includes symbiont, which is going to add another layer of complexity. And on top of that, we have something that you know now. That's the epigenome. That is, there's different marks that are going to be put in different parts of the genome of the coral that are going to be removed depending on different environmental cues or stressors. And that is going to make the coral behave in one way or the other. Okay, now, most of the studies about coral acclimatization or long-term adaptation to environmental stressors or to episodes of high temperature in the water. By the way, this is Elephantia getting rid of the symbionts during a bout of high temperatures, which is something that at the end of the day obviously is going to depend on the coral as well. But what's important in here is that most studies have been focused on symbiont. Different symbiont strains are going to provide a coral with higher tolerance to high temperatures. How symbionts are going to make it possible for the coral to keep living if they are not present and whatnot. But on the other hand, there's pretty much nothing known when it comes to the role of the host during those acclimatization responses. And there is where we are trying to contribute our knowledge or our research, right? So essentially, there are several emerging works linking changes in epigenetic marks and methylation in corals with responses to thermal stress as well as ocean acidification. Now, nutrient stress, thermal stress, those are our stressor models. We are trying to look at the epigenetic mechanisms in corals that are involved in the responses to these two drivers resulting from global change. Now, you're probably thinking, okay, so how is that informative for the purpose of restoration, of like trying to prevent those corals from dying? Well, because we're interested not in knowing those mechanisms, but also in knowing how can we use them as a way to select for coral colonies or coral individuals or groups of corals that are going to be able to better resist certain conditions. Now, that goes along with that thing I told you about these epigenetic marks being able or being transmitted transgeneration. Okay? So, now I'm going to give the word to Javier, who's going to tell you a little bit more about the specific research that we have been doing, and then at the end I'll be back to wrap up this whole thing up.
Yeah. Hi everyone. So, based on what Chema has been discussing so far, we're interested in those two things, the thermal and nutrient stress, and how those change the way coral respond and how this can be used for management of it. So, the main idea here is that most of researchers think that are considering very strongly now that based only on genetic diversity or the speed of what we know as Darwinian adaptation or the classic concepts, corals have no chance to survive. But many other authors have been proposing that phenotypic plasticity could be a mechanism in which corals could acclimate. But phenotypic plasticity is not a simple concept. So normally the environment affects organisms in very different levels, and we are considering only inside the organism, right? So from the genome to in the case of coral, the microbiome, which is more complex than just the symbiosis with the zooxanthellae, we have the process here is very complex, and the interaction between those levels is complex as well. So our interest is just targeting these two levels, epigenome and genome, how they crosstalk and how they define how coral will respond to environmental stressor, in this case temperature and nutrient. Okay? So in order to do this, we have like three very general objectives that we projected for this first part of the study. Right? So first we want to quantify photosynthetic download and DNA damage. Why? Because this is the way we can have a feedback of what is the physiological response of the coral. So going into what we care more, which is the epigenetic part, we first want to analyze how the nitrogen pollution specifically is affecting DNA repair based on an epigenetic mechanism. We will know why. I will explain more why we choose these mechanisms. And the third, we're going to go to the classical one. So we're going to study how DNA methylation is going to change under stress and how this DNA methylation is going to affect the physiological response and the possible acclimatization response of the coral, or even adaptation. We'll get there. So for this, in order to do this, we developed two very simple experiments. And they give us some preliminary data to respond to bigger hypotheses. So the first experiment was done in the Florida Keys with Acropora cervicalis. We exposed 144 fragments in the field to three treatments: control, nitrogen, and nitrogen phosphorus. So we took this down after 35 days, and we collect every week for small fragments to compare and do the molecular analysis. So the second experiment that was very similar, but in a way more simple scale. We did it on a microcosm, on a mesocosm setting in Morea with Acropora hyacinthus. In this case, we only used nitrogen and control, and we did like 4 replicates, so it was a very small experiment. So we developed 3 hypotheses based on the things we think can be happening here. But the hypotheses are way bigger than the experiment we have so far. We are planning more experiments to conclude the hypothesis, but we have some results that we want to share because they give us an idea that we are not that far from reality. So the first hypothesis is this: we are anticipating that exposure to neutral thermal stress will increase photosynthetic rates, then will increase oxidative damage and increase the DNA damage in both the coral and the symbiont cell. So the first part, what we have so far is we measure symbiont density on corals exposed to both nutrient regimes. And we found something very interesting, which is after just 24 hours, you have a positive increase in density, a very fast increase in density of the symbiont in both exposures. So, based on this, we're expecting that fast increase is going to affect the oxidative, is going to generate oxidative damage, changing the way these oxyanteli do the photosynthesis and the way its DNA and the coral DNA gets damaged. So, what's the next step? We need to check that. We need to do photobiology and oxidative stress and check the DNA damage. The problem with this is that we need to do this in situ. We cannot do it, for example, we collect before. So that's one of the objectives of our new next experiment. But in this, we found something very interesting for us, is that when you expose the coral just to nitrogen after several weeks, you see this. What's going on there? So we have a reduction in the symbiont density under the level of the controls and the everything else. So we think that this could be related with this, let's say, model Wilhelm proposed several years ago, when he said that it could be happening phosphorus starvation syndrome. In this case, when you expose a zooxanthellae population to increased nitrogen, they're going to tend to grow, right? They have nutrients, they're going to grow, but phosphorus become limited. But when this happens, then there are a lot of physiological changes inside the zooxanthellae. Basically, they start using phosphorus for the lipidic membranes. They change that in order to save phosphorus. And this creates a lot of physiological changes of the zooxanthellae that at the end are going to affect, to generate more oxidative damage. But what's going on with the coral? What's going on in the host? Is this going to affect the host, the phosphorus? I know the increase of oxidative damage is going to affect the host, generating DNA damage. However, we don't know what are the mechanisms coral have to deal with DNA damage. So one of the most generalized mechanisms in the eukaryotes is this one, the H2AX phosphorylation mechanism. Is one of the most generalized and probably the most important DNA repair mechanism. And as the name says, is depending on phosphorus for activation. So what will happen if phosphorus starvation is affecting also the host? Then you will have an extra stress. Not only that you are having an increased oxidative damage, is that you are being impaired, your capacity to repair your DNA. So, basically, we have our second hypothesis, which is exactly that. Phosphorus starvation is going to limit the functionality of this mechanism. So, the first thing here will be, is this mechanism present in corals? Well, apparently yes. So using our methods to isolate keystone that we use for several other invertebrates and using commercially available antibodies designed for humans, rats, and stuff like that, we were able to identify that H3X is indeed present in coral and actually it's being phosphorylated. So when we checked this on our samples from exposure, we found that after just 1 day of exposure, you see an increase in H2AX phosphorylation on both nutrient-rich treatments. That means that it is responding, this mechanism is responding to an increased DNA damage or increased oxidative stress. But after 3 days on the just nitrogen, on the nitrogen-only experiment, you see a reduction from controls of the H2AX phosphorylation. That means that this mechanism is being impaired, or apparently is being impaired by this problem. So what's next? We need to finish our data analysis for this big experiment on the keys, and we also have planned to do another experiment to target this probably with more specific antibodies to make sure that what we're getting is specifically this. So our last hypothesis is about, as I said, DNA methylation. But in this case, it's a very complex hypothesis, so we put it into 3 main things we need to address in order to respond. First one of our core ideas is our environmental condition influences DNA methylation. Okay, we know from other papers that apparently yes, but we need to prove it ourselves. So we checked DNA methylation on our experiment, and we found a reduction of the global methylation after just 24 hours. Again, we need to complete this data set and figure out exactly how long this takes and how long the response remains on the coral. But then this takes us to another part of this hypothesis, is that those modifications in DNA methylation influencing later physiological response can we be able to correlate methylation changes with changes in gene expression and transcriptomes? So that's the next step. We want to target DNA methylation in specific genome regions and then try to relate that with a group of genes that are going to be expressed under stress or non-stress condition. And even more important, after you have one exposure and you recover, what's going to happen with those genes in the next step? Is DNA methylation actually enhancing the response of the corals in a future exposure? That's something we want to address, and we think this is one of the most important things in order to inform management. So, in summary,
I'll be the one giving you the wrap up of this thing. So, well, basically, what you can see is that we are not dealing very much in terms of pictures of corals, but instead of that, we are things with photos and like proteins coming and going. But I guess that the take-home messaging here is that every time you see a coral dying, every time you see bleaching, obviously it's the effect of the environment, but also the interaction of the environment with the genetic and epigenetic ability of that organism to respond to that stressor. Now, if we have organisms that for some reason are more resistant to those environmental stressors, those organisms, their offspring, is probably likely to be resistant as well. Now, what we are trying to get across in this case NSF, if NSF is still alive after like this transition period in the White House, is that, basically what we like to do is essentially not just studying the mechanisms, but go to the field, get corals from the field, take those corals to the lab and expose them to thermal and nutrient stress. Measure, the response for a period of 1 month, the level of epigenetic and genetics and also the level of symbionts, and then return those corals to the reef for 1 year. And then take them back to the lab, do the same all over again, and look what's happening there. I mean, what we anticipate is that we're going to have naive corals that are going to be affected probably by those stressors. But once we do that, once that we expose those corals for the first time to these stressors, they are going to become what we have defined as preconditioned. And therefore, that means that they are going to remember that they are going to have an epigenetic memory that is going to make those response mechanisms more readily available next time they are in contact with this kind of stress. And therefore, they're going to be more resistant to those conditions. Now, that obviously opens many different directions. I'm not going to go into the mechanistic, genetic, epigenetic level, but there's definitely a lot of work in there. But probably the most important thing is, okay, so can these epigenetic marks and the genes that they are regulating be used as markers to identify coral colonies that are going to be more resistant to the effects of global change? That is our overarching hypothesis here. Since we are working with those marks, and those marks are somehow going to be transmitted or can be perpetuated in the population, we believe that they might constitute very good candidates for that. Now, the last thing that we need to do is acknowledge our collaborators, the group of rookie pilots at the University of California, Santa Barbara. They are the coral guys. We are sort of wannabes. Also, Dr. Simrox at the University of Washington, they are more interested in studying DNA methylation and genetic marine invertebrates. And in my lab, in addition to corals, we're also interested in a broader range of marine invertebrates, but also focus on this genetic mechanism. Well, Javier is really busy, is taking the lead in Gilmore, so he will be glad to ask any questions. Thank you very much.
So I was just wondering how do you answer to that? Because it almost feel like corals here are already exposed to that stress frequently enough to probably show some sort of epigenetic, right, some change in the narrative. So and I guess they're still dying. So, and I work on acclimatizations too, so I'm kind of probably throwing this question at you to see what you tell me.
Yeah, so I'm not a coral person, but I've been involved with this research lately, and I've seen that even though they have been, let's say, acclimated to these conditions before, the conditions are getting worse and worse. So the rate at which corals are being exposed to new conditions, it's faster than at least the way we can see that all of them are adapted. But since the last presentation we had before lunch, she was showing that there are colonies that actually are resistant to bleaching on a very fine scale. I mean that this coral can be more resistant than the neighbor that is 2 meters away. So what's going on there? What has that colony that is different to the other one? So epigenetic mechanisms are faster to occur, say, because you can trigger it by environmental condition, but we need to know which one of those are the ones that persist and help the coral to the next step. So what I've heard about coral people around is that bleaching is not very common in Florida till very soon, right? So coral were responding well to that temperature increase until now. And then we're having an extra increase, an extra problem. So Ben was talking about, inland water coming into the reef. If we are able to quantify not only temperature and nutrient by separate, and even, pH of the water by separate, we can put that together, that will be our response. We talk about bleaching as is only caused by temperature. That's probably the main driver, but you have a lot of stressors that are going to condition a coral to respond in a different way to bleaching or to temperature, to nutrient, or many things. I think that's our goal. Try to figure out what's going on at the physiological level of the coral, how many different things are going to be working together. Another problem we have is now all the studies they have been doing in the microbiome. So most of the bacteria and viruses generating diseases in coral are not weird things, are things that belong to the microbiome of the coral. So those became, a disease because of a condition the coral has. So it's a mechanism on the physiology of the coral which is making those bacteria to generate a disease. So I think it's very complex. That's why we try to target it at the most basic level, to try to find an explanation we can go up and try to explain more. But yeah, the acclimatization process, I think, is very complex, especially here in South Florida where you have coastal development everywhere. There is no place where you can go and find no 25 holes. So
I guess that one of the main reasons why we would like to develop that study in Morea instead of doing it here is because of the distinct conditions in the reef there.
you can isolate effects.
But the thing is that obviously if any of you folks who is interested in incorporating this kind of aspect to your ongoing work, we are looking for, actually, we were doing some stuff here in Florida Keys, but we are not doing it anymore because the people who was doing it here is not doing anymore. So essentially we're looking for contacts that are interested in sort of incorporating this kind of work to their portfolio. So if you are interested in that stuff.
So remind me about the design of your experiment. You had a control, elevated phosphorus, and elevated temperature, and those are the 3 treatments?
No. We just, at this point, all our experiments are being based only on nutrients. So we are not manipulating temperature yet. That's our interest. So that's the next thing we want to do. We try to do like a basic level, just one stressor. So we prove or we check that something's going on, and then we add temperature. So we're going to, and the experiment we're planning to do in Morea, that one will include both things. So how nutrient and temperature, or nutrient and temperature separate, could affect these mechanisms.
Okay, that's where I was going with that to address your question. Is that oftentimes what we see in terms of these proteomic responses are multiplicative effects, and be prepared for the responses to be orders of magnitude. Okay. So I think we think of these cumulative or different stressors impacting the coral, that they just add up, and that the coral is paying for it on the scale of like dollar bills. But they don't add up, they multiply. So instead of 2 plus 3 equals 5, it's 2 times 3 equals 6. Then the coral isn't paying 6, it's paying 6 orders of magnitude. So now you're talking about millions of units of stress on the coral. And so that's why if you do it as an experiment, I would really encourage you to do the full factorial, because that's what gives it a pop.
Yeah, that's what we are planning to do. But actually, over that, we found something very, very interesting for me. That was we were just with oysters, and when you check this marker H2AX phosphorylation in oyster, you find nothing in some oysters. I mean that those oysters have no response whatsoever to DNA damage. The coral doesn't have that. So even control corals have a high amount of H2AX phosphorylation going on. That means that coral host is dealing with a lot of DNA damage on a regular basis. So that means that coral is, all that idea of the symbiosis being beneficial for those, for both, being very happy situation. At least at the physiological point of view is not. Coral is paying a high cost for having a symbiote being inside. So if you add any kind of factor that is going to affect how the physiology of the symbiote or the coral, at any level is going to affect the physiology of the cell, of the coral, because it's the one with the cell inside. So.
Yeah, you're looking at sort of the last line of defense too, because there are all these defenses against the radical oxygen species. To start with superoxide dismutases and all sorts of other peroxidases. So it has to leak through those defenses before it gets to that.
But apparently it leaks very easily, because one of the mechanisms coral do is having a pigment to limit the amount of light that gets to the stuff. So imagine that you know that that's toxic, and then you try to cover it to limit the amount of toxicity it generates. So that means that normal mechanism that we have to deal with oxidative stress, they are not enough for corals. You need to have other mechanisms in order to control them. And repair mechanisms apparently are active all the time. And that's something I don't know, but I don't think it's common in any other organism.
Well, that's my other suggestion is to look at photooxidative stress. So you can do experiments duplicating the light and the dark. And that can help you get this mechanism a little bit faster.
That's one of the ideas. We are doing our experiment on Morea on a mesoconstant setting. So we are going to control everything, and we're going to measure everything. Because even if we try to limit as much as we can the factors influencing, for example, something we have to account is the amount of microbes that is in the water. So if we change just that, just the level of microbes in the water, then we are introducing another stress that we are not accounting for. So, yeah. So, thank you very much.